Good morning, everyone. It's a very great pleasure to be able to start the section on emotion by acknowledging the very many contributions that I have received from Larry Weiskrantz. I was an undergraduate at Cambridge, and he lectured to us in Part 2 Psychology. And I learnt a tremendous amount from Larry about a very wide range of topics. One of them was emotion, another one was memory, and another one was vision and inferior temporal cortex, which he was pioneering at the time. And all of those things have turned out to be the focus of my career. So I owe a tremendous amount to Larry Weiskrantz. Now, I'm particularly interested not only in psychology, but also in what Larry is interested in, the neuroscience underpinnings. And it's quite remarkable that in Larry's lifetime, we have been able to go from understanding very little about the brain to now almost understanding some of the computational principles of how these wonderful things, our behavior, emotions, and so on, might be produced. This is a new book, which I've just finished, which I was hoping to be able to present to Larry Weiskrantz today, but the printing won't be ready until the weekend, when the book will be launched at the European Society of Neuroscience, but I'll make sure Larry has a copy soon. Anyway, that's to say that something remarkable has happened in our lifetimes, that we've gone from understanding almost nothing about the brain in terms of how it works to now having ideas about the principles by which it computes. Now I want to show a slide to summarize some of the very important contributions of Larry Weiskrantz. In his 1956 paper on the amygdala, which we've heard about, he drew attention to the reinforcing properties that might be being impaired. That is, that you perhaps could not learn about rewards and punishers, and that might contribute to the emotional changes. This turns out to be a very, very important concept, that rewards and punishers are something to do with emotion. And this was one of the conclusions of his 1956 paper. Then he edited a book in which he wrote many chapters called Analysis of Behavioral Change. And in that he wrote an article on emotion, again, which was very seminal. He emphasized respondents, that is, things like conditioned responses and so on, in that article, but the seed idea of rewards and punishers was present in that and has stood the test of time. Then Larry Weiskrantz went on to do some very interesting pharmacological research on non-reward. And he developed the idea with Victor Balzer that perhaps one could look for an antidepressant drug by seeing whether it reduced non-reward. Think of you not receiving a reward. It might make you sad. So that's a way into understanding depression. And his idea with Victor Balzer was to try to look for antidepressant substances by looking for those that would reduce non-reward. Again, a seminal idea. And then we all know about the more recent work on blindsight, which will have a considerable focus in this meeting. So Larry has made many contributions. Now, I'm an active scientist, so I want to go on to summarize what we now think about emotion, and then to give you some completely new data, a new theory of depression. But to emphasize that comes out of the seed ideas from Larry Weiskrantz. So my particular approach to emotion is to say that it's a state elicited by rewards and punishers. And I'm focusing on the very important evolutionary function of emotion. The hypothesis is that genes are specifying many rewards and punishers for us. Many things we like, such as food, and many things we do not like. And by specifying these goals for our action, they have a very efficient way to influence our behavior, because all we need to do is to learn any behavior, any instrumental action, to get the reward or avoid the punisher. So emotions, I'm saying, arise out of this very powerful evolutionary adaptation of genes to influence our behavior. When we don't get a reward, for example, we have an emotion. When we do get one of the goals, we have an emotion. And that's the sort of deep idea behind the approach to emotion that I take. Notice that motivation also can be explained in the same way. If the genes are specifying the goals, then you have to be able to want the goals. So motivation and emotion, according to this evolutionary theory, are opposite sides of wanting the goal and then getting it. And then the thing that we've added on top of this more recently is that your behavior can also be influenced by reasoning. So notice that what I've said about emotion is it's for your genes. But once I can reason, I can choose to do something that is not for the good of my genes. I can choose, for example, not to eat a fattening food because I want to have healthy nutrition. So what happens is that there may be two different systems that influence our behavior. The second one, the reasoning one, can be good for our phenotype, for our own bodies, and is quite distinct then from the selfish gene one, which is good effectively for reproduction and which produces emotion. And the very interesting thing is that there may be a conflict between these two systems because one has a long-term goal and the other one has a short-term goal. And what I would say about blind sight is it may often, in relation to emotion, be tapping into the emotional part of what I've talked about, the selfish gene part, and enabling us to see what's happening with that, whereas the explicit reasoning system does not have correct access in blind sight. Now, I want to emphasize, therefore, that there are lots of different outputs to behavior. So the second one down there, action-outcome learning, is the one that's so important in relation to goals, learning to obtain a goal. And the amygdala and orbitofrontal cortex are very important in that. Then on top of that, we have an explicit system, which I've described to you. And then underneath that, we have implicit habits, and then we have things like learned autonomic responses. So the crucial point I'm making there is there are many outputs to behavior, and they won't all agree with each other. They're all different computational systems in the brain, and that helps us to understand, I think, also part of what's happening in blind sight. Now, I've already talked to you about the functions of emotion, so the second one is the important function. By specifying goals, we can learn any behavior to obtain an emotion. And we can see that emotions might be produced, for example, by a reward, which would produce happiness, or by a punisher, which at the bottom would produce fear, or by, for example, the omission of a reward, which might produce sadness, or by the omission of a punisher, which might produce relief. So this is a start into understanding many types of emotion. But there are many other factors, which I've talked about in books, which enable us to produce a rich description of emotion. Now, the brain systems, which are especially important, which I've studied, include the orbitofrontal cortex. So the orbitofrontal cortex here receives from the what systems in the brain. What visual stimulus, what taste, what smell, what touch. And it puts all of those together, and in what I call the reward tier of processing, then forms representations which are multimodal, which then become mapped into reward space. So the reward value is represented at that stage of processing, but not at any previous stage. And for Joe Ledoux, who's here, I think what happens in rats is very different to that. I think that's what is called the what system here does have hedonics in it in rodents, but much less in primates and humans. Now here's a little bit of the empirical type of discovery that we made. We discovered one of the principles of every reward system, I think, that if you have, in this case, a neuron in the macaque orbitofrontal cortex with a high firing rate to, say, the taste of glucose, then if you feed glucose to satiety, the neuron stops responding, but can still respond beautifully to BJ, black currant juice, fruit juice, which has not been fed to satiety. So a key principle of emotion is that reward systems show adaptation, punishment systems do not. If you have a punisher, you have to just remove it. Another key principle of the orbitofrontal cortex is that it's very good at learning. In this case, without going into the details, it can learn an association between a visual stimulus and a primary reinforcer, a taste, in one trial. And with practice, orbitofrontal cortex neurons reverse that in one trial, and they, in fact, have some special error neurons that help them to do that. So that's a very important principle of orbitofrontal cortex function in primates, including humans, that may be less well developed in rodents. Now, we discovered face cells in the inferior temporal cortex, but we also discovered that that system projects into the orbitofrontal cortex. So the orbitofrontal cortex of the monkey also has beautiful cells that can respond to faces. Some of them respond to face identity. Some of them respond to face expression. And some of them are beautifully tuned to head motion. So, for example, if I do this and look at Larry Weiskrantz, such a neuron might respond. But notice that that is an emotional signal. If I turn towards someone and look at them, there's a signal that something might happen. So that sort of neuron is probably important in social behavior and is found in the orbitofrontal cortex. So that slide just summarizes that we know a lot about the orbitofrontal cortex at the neuronal level, with many rewards represented there and many non-rewards, error neurons. Now, taking this forward to humans, we know that there's a representation of the same type in the human orbitofrontal cortex, but it extends beyond that to even abstract rewards. So this was a study we did that showed that monetary reward is represented in the medial orbitofrontal cortex, and monetary loss, and this is an important point, just try and remember this for what follows, monetary loss is represented in the lateral orbitofrontal cortex. We also did a study with faces in which one was given a choice between two faces. And if one chose the correct face, a smile resulted. But on some trials, we did a reversal and a frown appeared. And we did imaging to find out what happened on the reversal trials when you did not get what you expect. In other words, when there's an error. And here's what we found, that in lateral orbitofrontal cortex, the little red ring there, we get a big activation just on those reversal trials when you have to change your behavior. So that's further evidence that lateral orbitofrontal cortex is involved in non-reward. And again, this just summarizes many fMRI studies in humans showing many different types of reward are represented in the medial orbitofrontal cortex. There's a reward map. Whereas in lateral orbitofrontal cortex, many punishers are represented. Now I want to introduce to you just the new theory of depression, which has followed from this research, so that you get something brand new, only just published. So starting with these hypotheses on emotion, I refer back to the discovery we made of single neurons in the orbitofrontal cortex that respond to non-reward. In this case, what you're expecting when you see a visual stimulus is to get a nice taste. You'll see in this slide that a neuron responds very vigorously on some trials, and those trials are the ones when the monkey licks, expecting to get fruit juice, but gets salt. So he has to change his behavior. So those are error neurons, a mismatch between what you expect and what you get. Now we can put together that finding with a lot of MRI evidence. So if we look at A, we see the evidence I've just shown you about non-reward in reversal for face expression, activating lateral orbitofrontal cortex. If you look at B, you will see a new study we've just completed with 1,709 participants of the stop signal task. And what we find is that this inferior convexity level, which you can see, the inferior convexity is strongly activated when you are about to make an action and then someone tells you stop. And then C, a recent study in the monkey by Matthew Rushworth's group, showed that in monkeys who have to change their behavior, you also get this fMRI indication of non-reward in the lateral orbitofrontal cortex, just published in Neuron recently. Now if we look at the hedonic map, you'll see the orbitofrontal cortex here in a ventral view. All those yellow numbers indicate different types of reward, medial orbitofrontal cortex. All the white numbers represent different types of punisher or non-reward. So there's a hedonic map, including non-reward, with non-reward in the human lateral orbitofrontal cortex. So here then is the new theory of depression. The theory is that non-reward, which can produce sadness, is being computed in the lateral orbitofrontal cortex, and that system may be too sensitive in depression. So it may get triggered into what we call an attractor state. The attractor state refers to the fact that the single neuron I showed you keeps firing for many seconds after the non-reward is received. So this is a short-term memory, a function of prefrontal cortex, implemented in your orbitofrontal cortex, which makes you respond for many seconds when you get a non-reward. And the theory is that that system is too sensitive in depression. Now, interestingly, in humans, that system can go into the explicit reasoning system that I discussed with you. And so you can put the non-reward into a reasoning system, which can then reason, this reward may never come back. And that system in humans, the language-based system, can then feed back down onto your lateral orbitofrontal cortex to do top-down modulation of it, to keep it in the sad state. So this is what I call a long loop approach to depression, where there's an interaction between a non-reward system in the lateral orbitofrontal cortex and an explicit reasoning system, all implemented by these attractors. Now, to test this approach, we've just completed an MRI study of resting state activity in 421 patients with depression and 488 controls. And with resting state functional connectivity, you can measure the correlations between different brain systems. And you can ask which parts of the brain are overconnected and which parts are underconnected. And here's the result, which is in review at present. So for those of you who know the brain, you will see that some voxels in the medial orbitofrontal cortex are responding differently in depressed people. There's a small collection in the lateral orbitofrontal cortex. Marco, if I had a pointer, I could point to that. You will also see that the temporal lobe overlying the hippocampus, also, that's great, thank you. You will also see that the temporal lobe here, overlying the hippocampus, has many altered voxels, part of the memory system. So those are where the voxels are. Here is the change of functional connectivity that we detected involving orbitofrontal cortex. So medial orbitofrontal cortex here, in this ventral view, has reduced functional connectivity here, blue, with the medial temporal lobe hippocampal memory system. What do I think that's doing? What I think is that in depression, you're thinking less about happy thoughts. You don't get that effect from the lateral orbitofrontal cortex region there. Its connectivity with memory systems is not different. So there's an imbalance towards less connectivity of the medial reward system with memory systems in depression. That's one major finding of that voxel level study. The other finding is that lateral orbitofrontal cortex there is over-connected to some brain systems. So one of the systems is a region in angular gyrus, which is probably related to language. Another one is to the precuneus medial parietal cortex, where there's a representation of self. And the third one is to temporal lobe visual cortical areas, such as inferotemporal cortex. And here, roughly, is what we think is happening. In depression, we think this over-connectivity to the precuneus, which is very prominent, is concerned with increased non-reward sad feelings about yourself. Precuneus seems to have a representation of self in humans. And this is one of the first things that happens in a depressed patient going into a phase of depression. Their self-esteem goes right down, and we're relating that to this over-connectivity. At the same time, they reason about their state, and we think that is the over-connectivity from the non-reward system to the language system. And then we think this input coming from inferior temporal cortex, which will bring in visual stimuli, is being interpreted as being too non-rewarding. So instead of going through the different slides that summarize the arguments, I think you have the understanding that we have different functional connectivity in depression, which arises now out of this theory of emotion and is related to it. So what I'm going to do is, I think in the interests of time, I'm going to simply summarize that the reduced functional connectivity of the medial orbitofrontal cortex implicated in reward with memory systems provides a new way of understanding how memory systems may be biased away from present events in depression. And the increased functional connectivity of the lateral orbitofrontal cortex provides a new way of thinking how non-reward systems may be overactive in depression. And I would say that we can only do this sort of analysis now with these modern methods. We needed a large number of patients because we're dealing with this vast number of correlations between pairs of voxels. And so in this big data approach with modern neuroscience, we're able to get significantly different functional connectivity when we have about 1,000 patients in a study. Now, I can't sort of not mention consciousness at a meeting like this. I will not elaborate my theory. Many people have their own theory. My theory is fairly close, I think, to Larry Weiskrantz's. Larry Weiskrantz often talks about reflection, and I'm saying that there's a linguistic system which monitors its own planning. And if there's an error in that planning, the language system then can help to correct its own syntactic planning. And I'm saying I can't imagine a system doing that operation without feeling like something. I can't prove that last point, so it's a plausibility argument, but that's my theory of depression. Then I finish my presentation by returning to my tribute to Larry Weiskrantz and saying that many of the things that I've covered in my talk, the discoveries and so on, do reflect the inspiration, the wonderful inspiration that I've had over many years from Larry Weiskrantz. I'll stop there. Marco, you have to decide whether there's time for questions or whether you go on to the next.
So that's what I was saying. So it was planned to have a round table at the end of the 3 talks that compose the session, but we are doing well with time. So if there is a couple of short questions now, and maybe we can spare some time for longer discussion later in the round table, it's fine. Larry has a question.
Standing on the shoulders of giants, Cruver and Busey first made very large temporal lobe lesions and included the medial frontal. And various people, mainly Mort Mishkin and the Wilsons, have tried to disentangle subdivisions of that system. And it's good to be brought back to, that's how I got onto the amygdala. And that's why Mort Mishkin and others got onto medial temporal lobe lesions and so forth.
And let me say, Larry, in response to that, that in a way, the way I see the history of neuropsychology is that Pribram had 2 sort of very influential students, you and Mort Mishkin, and in a way, the 2 of you have seeded 2 sort of vast areas of neuropsychology. Yeah. Thank you, Larry.
Time for another question, or? No question? So we keep general discussion later. Thank you, Edmund Rose.